This is Lev and his friend Pheny. They help people stop having seizures. Lev has pretentious parents that wanted to seem so smart, so they named him Levetiracetam or Levetiracetam, or I don't know, they just love correcting other people's pronunciations. Pheny is short for phenytoin, after her family's lucrative Boston business, the Phenytoin Company. You may have heard a lot about the two of them recently. Their seizure-stopping powers were compared in two large randomized controlled trials, ConSEPT and EcLiPSE. Everyone thought Lev would dust Pheny in the competition. He is so new and fresh on the scene, and is even good-looking on his side effect profile. Unfortunately for all his adoring fans, the competition was much closer than expected, and the race officials did not have timing chips accurate enough to detect whether Lev or Pheny stopped more seizures. That, however, did not stop the media frenzy from claiming that Lev was equal to Pheny in stopping convulsive status epilepticus. Front page news on Up to Date proclaimed, new data from the two adequately powered open-labeled randomized trials ConSEPT and EcLiPSE suggest that Lev is equally safe and effective compared with Pheny. It is true that both trials failed to detect a difference between Lev and Pheny. In ConSEPT, Lev by himself stopped the seizure activity in 43% of the patients that failed to respond to two benzodiazepines, versus Pheny by herself stopping it in 46%, for a p-value of 0.65. In EcLiPSE, Lev and Pheny stopped the seizure activity in about 63% of the patients for a p-value of 0.87. This makes it tempting to claim that there is no difference between the two drugs. However, this is a common logical fallacy. This is accepting the null hypothesis. There likely is a difference between two drugs with different mechanisms of action, pharmacokinetics, and administration. That difference, however, may not be clinically relevant and not worth detecting. For example, if you run an equivalence trial on two drugs where the seizure cessation rate truly differs by 1%, say 61% versus 60, and you really want to find that difference, the sample size would need to be roughly 50,000 patients in each arm to be adequately powered. Therefore, the goal is to establish the largest acceptable difference that clinicians are willing to forfeit for the advantage of a better side effect profile, ease of administration, monitoring, and cost. That acceptable difference is a two-sided delta in equivalency trials or a one-sided delta in a non-inferiority trial. Since we care more about whether or not Lev is acceptably worse than Pheny, we'll focus on a one-sided delta. I'll come back to this, but let's first look at some data. Here is a forest plot of the outcomes in ConSEPT and EcLiPSE related to clinical seizure cessation rates. I included both the intention to treat and modified intention to treat analyses. Due to obvious logistical and safety concerns, randomization in both trials occurred prior to knowing which individuals would respond to the second benzodiazepine. The second-line agent had to be ready to go once the non-responders to benzos revealed themselves. Therefore, you'll notice there is a bump in absolute efficacy in the intention to treat analysis, because it included patients that responded to the second benzo without ever receiving Lev or Pheny. Any graph I show will include both the intention to treat and the modified intention to treat, but I feel the modified intention to treat is a more accurate reflection of efficacy despite the increased risk of bias. The trials had several differences, but perhaps the most notable was how ConSEPT's treatment algorithm differed from EcLiPSE's. ConSEPT's investigators pre-specified that if the patient still had seizure activity 5 minutes after completing the infusion of the first second-line agent, then the other study drug was given. The pre-specification in ConSEPT created a notable difference in the number of patients that received two second-line agents. In ConSEPT, 44% in the Pheny arm and 48% in the Lev arm received two second-line agents. The seizure cessation rate after one or two study drugs was 74% and 67% in the modified intention to treat analysis, respectively. In EcLiPSE, only 3% in the Pheny arm and 9% in the Lev arm received two second-line agents. Yet the seizure cessation rate after 1 or 2 second-line agents was 64% and 69% respectively. Therefore, a sizable proportion of the dramatic benefit of 2 second-line agents seen in ConSEPT might be a quirk of the algorithm rather than a true benefit. Perhaps this is because the physicians were not contemplating rapid sequence intubation as early in the algorithm and may have altered their clinical assessment and management. Whatever the reason, it's important to keep this difference in mind as I discuss and combine the data of the two trials. It is another reason why I am slightly more interested in the modified intention to treat analysis that looked at the proportion of seizures stopped with only 1 second-line agent. The data suggests that if you have 100 children that do not respond to benzodiazepines, about 50 to 60 will respond to 1 second-line agent. An additional second-line agent will only help about maybe 10 more kids while leaving 30 to 40 to continue to have seizure activity before proceeding to rapid sequence intubation. Whether that marginal difference outweighs prolonging status in 30 to 40 children is debatable, but that's for another video. Now let's get back to the real reason I'm making this video. Claims of equivalence from a non-significant result in a superiority trial. The burden of proof in the traditional superiority trial is squarely on the hypothesis claiming a difference. A significant result establishes a difference, where a non-significant result merely means equivalency cannot be ruled out. In order to claim something like non-inferiority, you have to flip the burden of proof on its head. Although definitionally awkward, the null hypothesis in a non-inferiority trial claims a difference, and the burden of proof rests on those claiming a treatment is no worse than some predefined delta. For the sake of exploration, let's say a 10% non-inferiority delta is an acceptable difference for Lev because of all his advantages. We'll discuss the trouble of creating a predefined delta and why you shouldn't make post hoc deltas in a bit, but humor the 10% non-inferiority delta for now. Here you can see a delta of 10% drawn amongst the various outcomes with both their modified intention to treat and intention to treat analyses. Only 3 would be interpreted as non-inferior. The 2 I am most interested in are the modified intention to treat of 1 second-line agent, and both are inconclusive. If you combine the data, the modified intention to treat of 1 second-line agent is still inconclusive. There is, however, something very deceiving about both these forest plots. That is, they are both woefully underpowered to detect a 10% non-inferiority delta. Here's the equation to calculate the sample size that is required for a non-inferiority trial. To calculate a sample size in the superiority trials of ConSEPT and EcLiPSE, the investigators hypothesized that Pheny has a success rate of 60% and Lev has a rate of 80% and 75% respectively. However, if you go through the studies the authors cite for their estimates, you'll realize the data available to them was all over the map and unreliable. There is considerable doubt that Lev is actually better. I'm making a short video that does a quick overview on the studies they use for their estimates, so you can check back in the future for a link in the description about those studies. Now, you could be super shifty and plug the hypothesized effect sizes from the superiority analysis into the equation above and show that the trials were more than adequately powered to detect a non-inferiority delta of 10%, with a two-sided confidence interval of 95%. ConSEPT would have only required 35 in each arm and EcLiPSE 54 in each arm to achieve a power of 80%. The reason is that your effect size from your hypothesis is so large now. That, however, is what I call trying to have your baby shark insanity too. If you get a non-significant result in your superiority analysis, then it is pretty dodgy to flip to a non-inferiority hypothesis without an appropriately predefined effect size. The burden of proof is turned on its head, as you are asking a different question. If the investigators wanted to reflex to a non-inferiority trial, they could have predefined a hypothesis of equal cessation rates, say 60% for both, meaning 0% difference. That is a generous hypothesis for a non-inferiority analysis, as the hypothesis normally assumes the experimental drug is worse, but not worse than some margin. Creating two different predefined effect size hypotheses may make some uneasy, but imagine if you did not have two hypotheses for the study. Think about what you would be saying. I hypothesize that Lev is 20% better than Pheny, and not worse than 10%. Duh. Being better or worse are mutually exclusive. Either Lev is better, or not worse than a certain margin, not both. If we plug an effect size of 60% for each drug into our sample size calculation, we would need 377 in each arm to adequately power a trial with a 10% non-inferiority delta. Based on the number of participants in the ConSEPT trial, it would have only been powered to roughly 35%, which means clinicians can only be 35% sure that a two-sided 95% confidence interval will exclude a difference of more than 10% in favor of Pheny. In EcLiPSE, the power would reach between 40 and 50%, depending on whether the analysis is modified intention to treat or intention to treat. Only when I reluctantly combine the data from both trials, the intention to treat analyses become reasonably powered at 70%. I invite you to play around with the data and explore why combining the data from these trials can be misleading. That said, I'm going to venture to guess the authors of the up-to-date article would not be comfortable with a conclusion of equivalence based on such low power. Remember from Stats 101, the problem with underpowered studies is twofold. Detecting true positives is less likely. At the same time, the false discovery rate increases when the significance level is held constant. When researchers get a statistically significant result, it is even more likely to be a false positive in a low-powered study. Now to the problem with creating a non-inferiority delta. All these sample size and power calculations were done under the assumption that a 10% non-inferiority margin is appropriate. Because the investigators of ConSEPT and EcLiPSE designed a superiority trial, neither provided a benefits versus harm analysis for a non-inferiority delta. So what is an appropriate margin for those claiming equivalence? A 10% difference in efficacy seems sizable when considering the pretrial hypothesized cessation efficacy for phenytoin was roughly 60%. Perhaps the delta should be smaller. In this video, I will not even attempt to defend any delta. The burden of justification rests with those making the claim of equivalency, and that's where we run into problems. Based on systematic reviews of non-inferiority trials, I think that the design and interpretation of many of them are shadier than an Armstrong doping program conducted at the bottom of the Marianas Trench on a cloudy day in the middle of winter. Maybe that is just me being optimistic, but it seems that a considerable amount of bias is creeping into the determination of non-inferiority deltas. I am leery when two reviews of about 400 non-inferiority trials found the comparator to be non-inferior in over 90% of the trials. Another study limited itself to high-impact journals such as the BMJ, Lancet, and the New England Journal of Medicine, and of 168 studies, 79% claimed non-inferiority, but less than half of these studies provided justification, and even when provided, the most common justification was an ambiguously worded clinical argument without details. A similar study found that only 2% of 182 trials met criteria for inferiority. Either researchers are so good at guessing which therapies are going to be non-inferior that the resources devoted to these trials is almost wasteful, or the deltas are being heavily biased and will lead to medical reversals and patient harms. My guess is the latter. From a purely trial design standpoint, it seems somewhat uncomfortable that the newer drug always has an advantage in non-inferiority interpretations. Your conclusions about Pheny in comparison to Lev should not change based on the whims of historical discovery. Here are two forest plots. Look at how the conclusions could theoretically change if Lev was discovered first. This unfortunate circumstance can be even more extreme if you follow the interpretation guidance published in JAMA. If you flip the historical discovery of example 1, it can become number 4, meaning that a drug could go from a superior interpretation to a non-inferior interpretation based on flukes of chronological discovery. older drugs also have the disadvantage of having a longer time to accrue a long list of adverse side effects that can unfairly bias the delta towards the drug we have less experience with. IV Lev wasn't available until 2006. Pheny was approved for use in treating seizures in 1953. Lastly, over time, a phenomenon known as biocreep can occur, which is when non-inferiority claims stack on each other with each new drug to a point where the latest and greatest drug is far inferior to the original control. Phew! This is a long video and I could go on about non-inferiority trials, but neither ConSEPT nor EcLiPSE trials were or claimed to be non-inferiority trials. However, I felt this video was necessary to dissuade those claiming equivalency without realizing the numerous assumptions they have to make and the mental gymnastics they have to perform to claim equivalency. In actuality, I really enjoyed reading these two trials when they first came out. It's unfortunate that they have been misinterpreted. I think they have stimulated some interesting debates, such as about the amount of time we are willing to sacrifice for a greater proportion of convulsive status to be stopped before rapid sequence intubation, or whether we need to revisit some older forgotten drugs, or some have even proposed moving rapid sequence intubation up in the algorithm. Perhaps just as exciting is that the SET trial, the US version of ConSEPT and EcLiPSE, will be releasing its results soon, so we can get an even clearer picture of how to best use current second-line agents in convulsive status. Until then, as always, thanks for watching and stay donkey.